we can get started with the recording. And yeah, so just for now, I think we are going to start running through the questions that I made. And if anyone else joins, they are certainly welcome to stop me and we can go over any questions you have. We can talk about the worksheet that we were working on through class. We can even go through the practice quiz if you want. Whatever, this is the time for you guys to learn as much as you can. So just to get started, let's see. I am not sharing my, 1 second, sorry. Sorry about that. So getting started with the questions, this is week 3, and we pretty much just did chapter 3 this week. We definitely started on chapter 4, but I think we can leave that for once we delve into it a bit more. So starting with question number 1. The color of a dragon's scales are coded for by a single gene, where green scales, S star, is dominant to the very rare golden scales. The last golden dragon that exists mates with a true breeding green dragon, and a single egg is created. What must be the genotypes of 2 dragons, and what phenotype might the egg exhibit when it hatches? So what are we even talking about right now? What is this? What's going on? Do we have any ideas? And for those of you who are watching, feel free to pause the video and try to take a guess for yourself. So this is what we would call a monohybrid cross. It's important because when you're in an exam, you might be asked, what's a monohybrid cross? Or it might say, you might get a question that asks, in a monohybrid cross, it's important to just know what you're dealing with. So what we're talking about right now is a monohybrid cross. That is where we have a single locus, a single gene that is being viewed, and we're seeing how it changes through generations by inheritance. So we can break this down. Mono, hybrid. The pen is doing it again. Mono obviously means 1. And that's because we are looking at a single gene. So in these 2 dragons that we're looking at, we have the last golden dragon, and they, because they are a golden dragon, obviously that is being coded for by the golden scale variant. What is this letter that we're looking at? And why is there a difference between green scales and golden scales? And that's because these are what we call, once again, alleles. Different variants of a gene, that gene being the gene that codes for the scale color in dragons. So we have these alleles. The last golden dragon exhibits this genotype. Because, hold on, let me back up. So we know that green scales are dominant to golden scales. And so in order for this golden dragon to be golden, they, as we can infer, have a homozygous recessive genotype. And they mate with a true breeding green dragon. What does it mean to be true breeding? We talked about it a little bit in class, but true breeding refers to homo versus heterozygosity. So we see that green dragons are green because of this allele, S star. So we know that in order to be a green dragon, we have to have at least 1 S star allele. But the fact that we know that this dragon, which is green, is true breeding means that its genotype is made entirely of one kind of allele. So this is its genotype. And any green dragon could also be, they could have this genotype and still be green because the green allele dominates the golden allele. But because we know that this dragon is true breeding, it has to have this genotype. So when you cross these two individuals, this is our monohybrid cross because we're only looking at one locus, one gene, which is the dragon scale color gene. We can very evidently see what the offspring is going to be because, sorry, one second, because this golden dragon can only create one kind of allele. They only have one kind of allele to then pass on to their offspring, and that is the golden scale color allele. And similarly, the green dragon only has the S star allele to give to any of their offspring. So they will contribute that allele to their offspring. And this is the only possible offspring that we can get from this specific cross. So these are the genotypes of the two dragons because of the recessive nature of the golden scale allele. We know that the last golden dragon is, or they possess this genotype. And because of the true breeding nature of the green dragon parent, they possess this genotype. So that was pretty easy. Oops. I forgot that it doesn't let me do that. Okay, so goodness gracious. Moving on to the second question. That was fairly elementary, so I don't doubt that we have that in the bag. Actually, let's see if there's anything I wanted to add to that. Don't look at this. Yep, monohybrid cross. That's something I didn't talk about. I thought I was forgetting something. So when we asked the questions, when we needed to answer this question, we asked what the genotypes of the dragons were, and we found out that they were SS for the golden dragon and S star, if it would let me draw the star, S star for the green parent. These are the genotypes because these are the letters that we use to represent the alleles, which in turn represent the genes that do something within an organism. But, you know, obviously when we look at ourselves in the mirror or when we look at cats on the street, when we look at our fellow human, we don't see the nucleotides. We see skin and we see hair and we see different lengths of limbs. And these visible traits that we see are what we call phenotypes. And so when we look at these dragons, the phenotype that we see is the color of their scales. So we have genes that code for specific traits that we can see. And pheno technically means shining. So you can think of like when something shines, we can only see, or something is only shining to us if we can see it, just like we see the visible traits that are coded for by our genes. That's kind of a lame analogy, but that's the etymology. Yeah, so moving on. That was a terrible transition, but that's okay. Moving on to question 2. It's related to question 1. Several hundreds of years, actually, let's get that going. Right, that's just going to be a nightmare. So keep in mind the result of this. We had an S star egg. That egg, yeah, that hatched from this cross between the two parents. So moving on to question 2, several hundreds of years after this egg has hatched, the dragon that comes from the egg becomes sexually mature and seeks out a mate of their own. They find a mate who is a carrier for the golden scale trait. When these two dragons are crossed, how likely is it that their egg's genotype will be homozygous recessive? So there's a lot of things in this question that we need to identify their meaning before we can truly answer the question. Just to start, what does it mean to be a carrier? We talked about it in class. A carrier is, it's easy to visualize for like a disease, for example. Let's say you have, let's say you're infected with influenza. So actually, no, that's a bad example. Let's say that you have cystic fibrosis. We can use an example that we are familiar with from class. And let's say that cystic fibrosis is genetic. And let's also say that it is only controlled by one gene, and there are two alleles that represent this gene. So because it is only controlled by one gene, we're only looking at one locus, the F locus. And because it is only controlled by two alleles, we have, two alleles representing it. Oh my God, what is happening, dude? Okay, sorry. Yeah, so this individual, oh, and assuming that cystic fibrosis, of course, is a recessive trait. I'm sorry, I'm fumbling this one. Looking at this individual right here, we see that they don't have any of the recessive alleles that would contribute to cystic fibrosis. This individual is homozygous dominant, and they have no cystic fibrosis allele. This individual has two cystic fibrosis alleles. They are all of the genetic information in their genome that has anything to do with cystic fibrosis is coded for by these alleles that make them susceptible or that cause them to have the traits that lead to cystic fibrosis. And so they are not, or excuse me, they're homozygous recessive, and they evidently display the traits associated with cystic fibrosis. But this individual here, they have one dominant allele, which means that they don't have any of the characteristics associated with cystic fibrosis, but they also have an allele that does code for those traits associated with cystic fibrosis. But because the one that doesn't dominates the one that does, they don't explicitly express a phenotype of cystic fibrosis, but they carry the allele and can possibly transmit it or give it via inheritance to their offspring. That was a really long-winded and terribly worded way to say that, they have one allele that codes for cystic fibrosis. So they have a mate. Going back to the dragon, we have a mate who is a carrier for the golden scale trait. So we know that they aren't a golden scaled individual, but they do have that allele. They do carry it. And we are considering this egg that we were talking about back in question 1, who also had this genotype because of the cross of their parents. And so we actually have 2 carriers crossing. And you can do the Punnett square for this, but we should know by now, we should have this well in hand, that this creates specific ratios of offspring when they cross. What does it mean to cross? Very simply, we have this X. So when we cross 2 things, it means that they mate and they combine their gametes that they share or that they contribute. And those gametes combine, they're crossed together to create an offspring. So when these 2 cross, there are a few possible combinations that can result. And the way that I think of either monohybrid or dihybrid crosses is that we have parents and they quite literally like pass on one of their alleles. This parent passes on the S star allele. This parent just happens to have passed on the S allele to their offspring. And so when these gametes combine after the parents cross, we get an individual who is S star S because of the alleles that were passed on. But it doesn't always have to be that way. It could be that the parent instead passed on this S star allele, and therefore offspring becomes S star S star, and you can do all those variations yourself. And so the possible combinations that we can get from a monohybrid cross are that this S combines with this S, giving us S star S. Or this S combines with this S, giving us S star S star. This one combining with this one, giving us SS, or this one combining with this one, giving us SS star. And so when we look at these, we see that we have one individual who has two S star alleles. So they are a homozygous dominant individual, which we know from the previous question. And we have one individual who is homozygous recessive. And we have two individuals that are heterozygous. They have one of each kind of allele, one star and one unstarred, one plain S allele. And so if we broke these down into their genotypes that we call them, we'd have one homozygous dominant to two heterozygotes to one homozygous recessive. And this 1 to 2 to 1 ratio is their genotypic ratio because it is their, oh my God, it is their genotypes that we're looking at. It is their genotypes that are similar. However, this genotypic ratio isn't always expressed as an explicit or differentiated phenotypic ratio because of dominance. So because the S star allele dominates the S allele, any heterozygous individual in this cross would show up as green. They'd have green scales. Obviously, a dominant individual with two star alleles would show up as green. So if any heterozygote is green and any homozygous dominant is green, that would be 3 of the 4 individuals being green. And one individual, this homozygous recessive, has no star alleles. They have both of their alleles are golden recessive, and therefore they show up as golden. This is very elementary, but I thought I should articulate it just in case. So how likely is it that their eggs would be homozygous recessive? 1 out of 4, because there are 4 possible outcomes, and only one of them is homozygous recessive. That was very poorly done. Yep, very simple. Didn't have to spend a lot of time on it, but we can move on. And this is something that we had a lot of practice with, but a lot of us still struggle with, and that's fine. Math it's not my strong suit. It's probably not a lot of yours. So it just takes a little bit of practice. And this is a really simple example that I think can help propel our ability to do probability in future questions. So if you toss a coin, what is the likelihood that it lands on heads? Assuming that, it's a normal coin that's evenly balanced or whatever. It's 50-50. That makes sense, right? It is likely that on 50% of coin tosses that the coin will land on heads. And so this is the likelihood that if you toss it once, it'll land on heads. In a single event, that event being the toss of a coin, this is the probability that it will show an outcome that you desire. However, if you keep tossing the coin over and over again, let's say 11 times, how likely is it that it would land on heads every single time? And these examples are often very difficult to penetrate, but if we can step back and think about it in a sensible way, it should never be a problem for us. So I think the key is realizing that if you need a specific outcome a whole bunch of times, it's much less likely to happen. Like if I were to walk into a field, how likely is it that I would get struck by lightning? Not very likely. How likely is it that if I walked into two different fields, I would get struck by lightning both times? Even less likely. So you can think of it as the more strict your requirements are, the more a specific event has to happen, the less likely it's going to be. And because we represent likelihoods as fractions, when we as decimals, when we multiply decimals together, the number becomes smaller, the probability becomes less. And so when we have more and more requirements, when we have to do something more and more times and get a specific outcome, we multiply those events, the probability of those events together. This pen is really giving me the business and I'm not a fan of it. So we multiply them together and that number becomes smaller. The probability that this happens twice is less. And so if we toss a coin, the base likelihood that it happens once is 0.5. Because, if something is a probability of 1, it is going to happen all 1 times, or 100% of the time. So if we flip a coin on half, I'm not going to keep articulating this. So if we toss it 11 times, it's just going to be 0.5 times 0.5 times 0.5, 11 times. And we can also write that as 0.5 to the 11 because we're math geniuses. And that gives us, I think I did the math earlier, but it's like 4.88 to the negative 4th power or something. Very simple example. I was just trying to word it better because it can be confusing. Probability isn't everyone's jam. And as an extension of this probability, let's revisit our previous example, thinking back to the dragons in a manner that's unrelated to the scale color genes that we were talking about earlier. The eggs that are laid by dragon parents can have different qualities. They can either radiate their own light, sparkle in sunlight, or reflect all light and look like, a spherical mirror. And any of these 3 egg types are just as likely as the other ones to occur when an egg is laid. It's just as likely that this happens as this one, as this one. And so if that is the case, what is the likelihood that any egg, specifically the egg that we got from question 2 up here, which was the cross between 2 heterozygotes, 2 carriers, which we remember that ratio of 1 to 2 to 1, where 1 was homozygous dominant, 1 was heterozygous, and 1 was homozygous recessive. So considering that cross, and now considering the different conditions that the egg could express, what is the likelihood that the egg will radiate light and then hatch a baby dragon with green scales? So just as we were talking about in question 3, if you want a specific outcome to happen multiple times, or you want multiple specific outcomes to happen at once, you're being more and more specific. And when you're more and more specific, it's less likely that those events will happen. And when that event is less likely to happen, the probability is reduced. And because we express probability as decimal numbers, that decimal number is going to reduce, or it's going to decrease. And the way that we decrease decimals is by multiplying them together. So in this case, the likelihood or the probability that we have an egg, the probability of a radiant egg, oh my God, sorry, I'm gonna figure this pen out someday. The probability of a radiant egg the probability that we get this condition is the probability that we have a radiant egg times the probability that we have a dragon with green scales. So that's very simple. If any of these 3 conditions are just as likely as the other one, it's like flipping a coin, but instead of 1 out of 2, since we have 3 options, it's 1 out of 3. Times the probability that the dragon is green. What is the probability that any dragon resulting from this cross is going to be green? Remember, green is our phenotype, and we infer phenotype from genotype. If one of them is homozygous dominant, one is heterozygous, and one is homozygous recessive, and we know that dominance creates green dragons, these 2 options have at least one dominant allele that represents the genotype. This one doesn't, and will therefore appear golden. So the probability is these 3 will be green, this one will not, and out of 4, we have 3. These 3 out of 4 will be green. So the probability of these 2 independent events happening together is their individual probabilities multiplied by each other. And 1/3 divided by 3/4 is some number that I forgot. Oh, I actually did the math, but it's somewhere else. $0.2475. All right, that's very sensible. So if we have the takeaway from these questions is, like, if we're trying to get a more and more complicated event to happen within a single event, like within a single birth of an egg, we want that egg to have 2 specific conditions. It's going to be less and less likely as we add more and more conditions. It's also going to be less and less likely as we reiterate several events to create the same conditions over and over. Like the coin toss, for example. Okay, enough of dragons. You are a ferret, and you love to dance around mischievously. and hunt for prairie dogs. Once you find a mate, you cross and produce 8 kits. That's a baby ferret. The tendency for mischief is coded for by a single gene, where mischievousness is recessive to docility, mischievousness being represented by the allele lowercase m and docility by uppercase M. If you are mischievous and your mate is docile, how likely is it that together you'll only have one mischievous kit? We can do this, okay? So what is this again? Actually, yeah, this is a monohybrid cross. And I guess some etymology that we can get out of the way that I kind of wanted to get to earlier, but it's very intuitive. But just in case, let's talk about it. What does it mean to be heterozygous versus homozygous? Pretend there's an O in there. So hetero means different. And we know from homologous chromosomes that homo means the same. Zygos refers to when you have two parents, for example, SS, S star, S star, that when they give one of their alleles within their gametes, because that's how genetic information is passed, we learned about meiosis, that these combine into their offspring. Oh, what is happening, dude? Into their offspring, which we know as a zygote. The zygote is the diploid cell that results from the combination of two haploids. And this zygote is what we all started off as, a single cell with some combination of our parents' DNA. And then from this single cell, we underwent mitosis and grew into an entire organism. That is what zygotes is referring to. So zygos, talking about the zygote, and this information within the zygote comes from our parents' genomes. So a homozygous genome, or excuse me, homozygous individuals have the same alleles, like S star versus SS, within their genome that they will give to a future zygote. And obviously heterozygotes will have two different alleles. So ferrets, hunting prairie dogs, mating. Let's do some stuff. So obviously we're looking at probability. How likely is it that something happens? And we explored a fancy equation that we can use to find this answer. So with the coin example that we had earlier, it was very simple, not very nuanced. We could just flip a coin, and every single time we were only looking for a specific, or for one specific outcome, and that was that the coin landed on heads. But not every series of events is going to have the same outcome, and sometimes we don't even want to experience the same outcome in every single event. So in this question, for example, we want to know about the probability of two different events. Think to yourself about what those might be, and that is the fact that we only want one mischievous kit out of 8. So if we only have one mischievous kit, then the opposite of that is having 7 docile kits. So these two are like opposing forces, and that is where we will jump into our probability. If you took biostatistics or AP in high school or whatever, you were probably exposed to an idea like this one: P plus Q equaling 1. P is the probability of one kind of event and Q is the probability of an antithetical event. So referring back to our heads and tails example, let's say that P is the probability of landing on heads, and Q, therefore, would be the opposite of that, landing on tails when you toss the coin. And the number of times that you toss the coin and get heads, plus the number of times that you toss the coin and get tails, equals 1, equals 100% of all results from the tossing of a coin. It's not possible to get anything but heads or tails. And in this situation, it's not possible to get, actually, let's talk about that. So we know our P's and Q's. So P and Q are probabilities. The probability of one specific event versus another, versus its opposite. So let's discover what these probabilities are. We know what the opposites are. The opposites are mischievous kits versus docile kits. So let's find out what these probabilities actually are. How do we do that? Obviously, we're going to look at a monohybrid cross. We talked about our alleles, and we even mentioned the cross. So you are mischievous little ferret, and we know that mischievousness is recessive. So in order for you to be mischievous at all, you have to be homozygous recessive, because if you had any, or at least one dominant allele, docile allele, then this would dominate your mischievous allele, and you would not express a mischievous personality, a mischievous phenotype. So we know that you have to be lowercase m, lowercase m. And now we look at your mate. It says your mate is docile. So we know that because they're docile, they have at least one dominant docility allele. But we've come across a problem. Because we know that docility dominates the recessive mischievous allele, you could be uppercase m, lowercase m, or uppercase m, uppercase m. Your mate could be either of these things and still be docile. We don't have any more information. So we're actually going to have to diverge. We're going to have to take two different pathways. Because your mate is docile and we don't know what their genotype is, we only know their phenotype of being docile. In order to find the probability, we're going to have to take both paths. So we might come up with two answers. So let's rewrite this. So let's explore the possibility that your mate was uppercase M, uppercase M. So when we make this cross between you and your mate, obviously you can only contribute a recessive M, and your mate can only contribute an uppercase M because that's all they have to give. So in this case, the offspring is uppercase M, lowercase m, or backward. And what do we notice? What is the phenotype of this offspring? The phenotype is that because they have at least one dominant allele, which in this case codes for a docile predisposition in a ferret, that means that this genotype corresponds to a docile phenotype. And because this is the only possible outcome due to the limited kinds of alleles that parents can contribute in this cross, 100% of the offspring will be docile. And so if 100% of the offspring are docile, then it stands to reason that 0% of offspring will be mischievous. And so we can kind of discard this possibility. It's not going to contribute to the answer anymore. So because we know that, let's now explore this one. We can keep that in mind, that the other cross would result in 0 mischievous individuals. We can keep that in mind for this answer. So let's explore this cross now. When you cross these individuals, you can once again only contribute a lowercase m. But, oops, didn't mean to do that. But your mate can either contribute dominant m or a recessive m. So when you combine those, you get dominant m, lowercase m. Actually, I'm going to be consistent with my colors. Dominant m, lowercase m. And now we, or you, I guess, contribute the only allele that you can, that is the lowercase m. So in this instance, this genotype corresponds to a docile phenotype, because, docile is dominant. This genotype corresponds to a mischievous phenotype because it's recessive, and therefore a homozygous recessive individual, like we see in this genotype, would give the mischievous phenotype. And because it's just as likely when we're going through meiosis that the parent gives either this allele or this allele, these two outcomes are just as likely as each other. So it's 50% likely that they end up mischievous and 50% likely that the result of this cross is docile. And so we have our P's and Q's. P in this instance is 0.5 and Q is 0.5. I'm going to say for this problem that P is the probability of an offspring being mischievous, and Q represents the probability of being docile. Hooray, so we have that out of the way, and it's very nice and clean, 50-50. So now that we know the probabilities, how can we put them together in a meaningful way based on the number of events that have to occur? Well, we know of a specific formula that we can use that will get us there. And that is the binomial expansion formula. I will rewrite it. It is such that the probability of a specific number of events happening is the number of events total, factorial, that's what the exclamation point is, it means factorial, divided by the number of occurrences of the event associated with the probability P, which we know as S, times the factorial of the number of events associated with, I'm actually going to use a different color, the number of events associated with the event Q. And the way that I keep these numbers straight is I just remember Q goes with T, and it's like you're calling someone a cutie. Q goes with T. So we know our P, we know our Q, we actually do know our T, and we know our S. So because we know that S is associated with P, and P is all about mischievousness, we want only one mischievous kit. So S is going to be 1. And because Q is the opposite of P, Q has to do with docility. Because we have 8 events overall and only one of them can be mischievous. That means that the other 7, by extension, have to be associated with docility. Oh, I didn't finish the equation. Let's do that. And this expression here is then multiplied by P, the probability of a specific event, to the power of the number of events, the number of times that event occurs, also multiplied by its antithesis, the opposite event to the power of the number of times that opposite event occurs. And so we can, because we have all of our numbers, we can very nicely and neatly just plug them in to this equation. And I also did the math for that earlier, and I don't want to retype it into my calculator. Oh yeah, so n is 8. We can also do this. 8 factorial divided by s is 1, because we only want one mischievous pup, times 7 factorial times, thankfully it's a nice even, or nice clean little 0.5 to the first power, times 0.7 to the 7th, or sorry, not 0.7, 0.5, because that is the probability of getting a docile kit from the cross MM with MM. And this gives us oops, that's messy. That gives us this number here, 0.03125. You know, that's kind of small. And that makes sense, as we were talking about earlier. When we have more and more specific requirements, you know, like out of 8 kits, only having one of them be a specific way, even though it's just as likely to be that way as it is to be the other way, being docile. As we become more and more specific, it becomes less and less likely. So we therefore get a small number like 0.03125. I think that was the answer. And this is the probability of only having one mischievous kit out of 8 total kits. And so there is confusion expressed about when to use this equation versus the nice, simple, like coin flipping equation, the specific probability very simply multiplied by other probabilities associated with different events. And that is because we only use this equation when we want a specific distribution of events, a specific combination. We only want one number, one event out of a predetermined number. We had previous examples with cystic fibrosis and albinism. So also look to those for examples. Excuse me, I'm gonna get a drink of water. Sorry, one second. Has anyone else joined our happy little meeting? Nope, just you and me. And that is A-okay. I hope you're having a nice day.
Yeah, I am.
And if you have any questions, please jump in. You would never be interrupting me. I'm here for you. So if no one has any questions, we can move on to number 6. I am growing some blackberries in my garden. A dihybrid cross of 2 blackberry flowers occurs, and I can see generational variation in the sour taste of the berries, represented by the letter T, and the number of druplets represented by the letter D on the berries. If sour taste dominates sweet taste, capital versus lowercase t, and an even number of druplets dominates an odd number, capital versus lowercase, what percentage of the offspring will have sour berries with an odd number of druplets? So where do we even start with this? This is a long paragraph style question, and what the heck? I don't know what I'm doing. It all relies on a single phrase. I'm sure you caught it. This one. What is a dihybrid cross? Take a second and answer it for yourself. But a dihybrid cross is what Mendel saw when he was looking at his peas. And we can break it down so that it's not this alien sounding word. We did it earlier with monohybrid. So di is a Greek prefix we know to mean two, and hybrid is, a combination, an amalgamation of two different things. For example, having uppercase versus lowercase m in the previous example. But this is a single hybrid. This is a monohybrid. A dihybrid is when we also look at another hybrid, one, two hybrids. And this is in genetics when we look at two different genes at two different loci. So dihybrid crosses are all about looking at two different genes and seeing how they're inherited. So dihybrid cross of two blackberry flowers. We know we're going to be doing the business of like AABB crossed with AABB, So we have our framework that we're going to use. We see generational variation. That is what Gregor Mendel saw with his peas. He noticed that they weren't all either yellow and round or green and wrinkly. Some of them are green and round, some of them are yellow and wrinkly. So we're looking at variation between different generations. And we know from the language of the question that sour taste is dominant to sweet taste. Maybe it's just the right side of my screen that doesn't like being written on. Sour, but I'm right-handed, so that's kind of hard. Sour and sweet. And then what color do I want to use? Sour and sweet. And then whether or not the berries have an even or odd number of druplets. Capital D is even, and it is dominant. Lowercase is odd and recessive. And a druplet on like a blackberry or a raspberry is when you're looking at the berry as a whole. Each of these like individual pockets is a druplet. And that's because rose fruits like peaches, plums, almonds, cherries, they're called drupes. They have a pit in the center. Raspberries and blackberries are a bunch of drupes all put together on a single little berry, which is called a multiple fruit. And so each individual little ball sphere of juicy goodness is called a druplet. So these are the phenotypes that occur relative to the genotype, or at least, the alleles and what they code for. So we have this. We know we're doing a dihybrid cross. Dihybrid between a TTDD is going to end up being, actually, let's be consistent with which one is first. It's going to be T, T, and D. Sorry, my handwriting is being kind of terrible. When we cross these together, this is our dihybrid cross. And this should look very familiar to you, and it is going to give us a very nice ratio, a nice even distribution of offspring that could possibly occur from this cross. And we've iterated in class several times that you don't really, if you're interested in saving time, you don't want to do the Punnett square for this cross. I was actually planning on doing the proof of it here so that from now on, we can believe in the ratio. That ratio is, I'm sure you remember, 9 to 3 to 3 to 1. Actually, do I want to do that? No, I don't. But suffice to say that whether or not this parent gives a specific gamete, because when it comes time to do meiosis, it's going to either give, this parent is either going to give this big one in combination with this big one, resulting in TD, or it'll give this big one in combination with the small d, resulting in capital T lowercase d. And the same, you can like FOIL out the rest of the possible gametes that result from that organism. And so it'll create different gametes that it will pass on, which will combine with a gamete from this parent and create our zygote that has some genotype, whatever. And when you do this with all of the possible gametes, you come up with a specific ratio such that nine of them will have dominant alleles, or at least one dominant allele at both loci. 3 of them will have a dominant allele at the first locus, and both recessive alleles, or 0 dominant alleles at the second locus. Another 3 of them will have 0 dominant alleles at the first locus. Oh my gosh. And at least 1 dominant allele at the second locus, and 1 of them will have no dominant alleles at the first locus and no dominant alleles at the second locus. This is the very familiar and easy to remember result of our dihybrid cross. And these are the genotypic results of that cross. So now that we know the genotypic results of the cross, we can start answering the question simply by associating a phenotype with that genotype. So let's talk about it. The answers are actually in the paragraph. We know that having a capital, or having at least 1 dominant allele for the T locus, the berry taste gene, whatever. Having a dominant allele at that locus makes you sour, because we have at least 1 dominant allele. And having a dominant allele at the druplet locus gives you an even number of druplets on your berry. So we can refer back. Capital T makes us sour, but we have no dominant druplet alleles, and therefore we are odd numbered in our berry druplets. And you can make up the rest, I'm sure. Sweet and even. And since we have no dominant alleles at either locus, we see the recessive phenotypes at both loci, sweet and odd. So now we can answer the question, now that we've matched up a specific phenotype to each genotype. What percentage of the offspring will have sour berries with an odd number? Sour and odd, boom. 3 out of all of these different times. 9 plus 3 plus 3 plus 1 is 16 total. So 3 of those 16 offspring, or 3/16 of any, how to word that? The answer is 3/16. Simple as that. So moving on to question 7, we're going to do a little bit of complication. Not necessarily, we're just going to make it a bit more involved. Okay, what is the likelihood that any of the offspring will have either sour berries with odd druplets or sour berries with even druplets? So let's keep this in mind. This probability was the probability of having sour berries with an odd number of druplets, 3/16. And so we want to know the likelihood that we have offspring with either sour berries with odd druplets or sour berries with even druplets. Or, wow, we haven't seen that one yet in this review, but we've seen it in our examples in class, and we know that an or statement in a probability question, it expands our options. Like, if your mom asks you to go to the store and get either apples or peaches, it's more likely that a store will have apples or peaches. I didn't say that well. The more options you have, the easier it is to satisfy the requirement. Your mom required one kind of fruit and you had 2 options. So it was more likely, or it becomes more likely as you are given less specificity, and therefore you increase the probability. And the way, because we express probability as decimals or fractions, the way that you increase the value of a decimal is by adding 2 of them together. So when we see this or condition, when we expand our possibilities or become less stringent with our requirements, we increase the value of the fraction or the decimal, and we increase the probability. So we have this or condition. So we can add our probabilities together. 3/16 was the probability of sour berries with odd druplets. We did that in the last question. And now the probability of sour berries with even druplets. We can find that right here in our nice little table of the dihybrid cross phenotypes. And we see sour and even, nice and plain. 9/16, or because of the randomness that organisms pass on gametes to possible offspring, 9/16 of the time they will express a certain phenotype. And in this case, that 9/16 is sour and even, so we can use it here for our probability. All right. So the probability that either event can happen is the probability that both individual events happen. Oh my God, I've been talking for a long time, I'm sorry. The probability that either of 2 events happen is the individual probability of either event added together. And in that case, we end up 3/16 plus 9/16 is 12/16, or 0.75. Whew. So this was pretty elementary. We had a lot of practice with it in class. But I thought that maybe exploring some different ways to say it, some less genetically inclined examples would make it a bit more palatable, or it could fill in the cracks of your understanding a little bit better. And you won't come across many more complicated examples than this. I don't think I think what you saw in class is just about the extent of what you will find on an exam. Oh, we have a new friend in the review. Hello. Um, we just finished up the questions, and I'm not sure what time it is. It is 2:10. We still have 50 minutes technically. Would you be interested in going through the practice quiz, or do you have any questions for me?
Actually, I do have a question.
Yes, what's up?
So can we go back to the worksheet, the review worksheet that you just did for a second?
Oh, yes, one second.
I'm sorry, I didn't want to interrupt you while you were doing the whole thing.
Oh, no, you are
We're in a
totally
flow.
welcome to interrupt me.
Yeah, I just thought I'd ask you after you're done.
Okay, sorry. In the future, you are absolutely welcome to interrupt me, and so I can clarify better in the moment and you don't miss the wording of what you want to say in your question. Yeah, you are absolutely fine either way. And I accidentally closed my browser, so I have to bring it back up.
I'm so sorry.
Oh, it's no problem. I'm sorry that I didn't stop for any questions in the meantime. And I can share the screen now. Awesome. What was your question? Which one?
So for question number 5, I don't know, it completely went over my head how you got big M and lower M as opposed to like big M and big M.
Oh, you weren't sure why I used only like one of the two options in the cross.
Yeah.
So it was kind of just the way that I built the question. I wrote these like 30 minutes ago, but I kind of made it ambiguous about what the cross even was in the first place. So we know that because being mischievous has to do with the recessive allele. In order to be mischievous at all, you have to be homozygous recessive. So we know that this is your genotype. However, the genotype of your mate is less clear cut. Your mate is simply described as being docile, and being docile because we know that docile is dominant, mischievousness is recessive to docility. Therefore, docility is dominant to mischievousness. Because your mate is only described as being docile, in that case, they only have to have a single dominant allele. And this means they can be either this way or this way.
Mhm.
And if we did this way, what kind of allele could they possibly give to any of their offspring?
Only the big M, so the all their offsprings would be heterozygous.
Exactly, because this you would have to give your lowercase m. That's all you have available to give in the first place. So all offspring would be heterozygous, and therefore all offspring would be what phenotype?
There would be the docile.
Exactly. And therefore, they would not contribute at all to being mischievous because it wouldn't be possible to create a mischievous kit from
Yeah.
this genotype.
Okay, so you just did like the heterozygous mate because there's a possibility of them having like a mischievous kit. That's why.
Exactly.
Oh, okay. I thought like there was some logic behind how you got that. I'm like, oh my God, did I miss that?
Oh no, I'm sorry. I should have articulated that better.
Yeah. Okay. That was the question that I had.
Yep.
Thank you.
You are so very welcome. So I don't really have anything else. If you'd like to explore some more stuff, we can definitely do that. We could talk about the worksheet that we did in class, or like I said, we can go over the practice quiz, whatever you're into.
I didn't do the practice quiz.
Okay, no problem. Do you want to?
Yeah.
Awesome, no problem. Let me log in first. And Daniel, did you happen to have a question?
Yeah, my question was like what we did last class with like the probability formula. I didn't really know like how to set that up and like the wordings of the questions.
Okay. Just one moment, please. Do you mean a question like this one?
Yeah,
Awesome. Do you want to just go through it step by step?
yeah, that'd be nice.
No problem at all. Okay, so what is the probability that the couple would have 7 kids, 3 with albinism and 4 normal? So right away, we can pick out that we're going to have to use that funky long equation, you know, that a probability, the probability of this like occurrence, 3 with albinism and 4 that are normal out of 7. We should be able to instantly pick out that we're going to have to use this equation. Because why? Why do you think so?
Because like there's different probabilities of different other ways that it could have gone.
Exactly. Yeah. We're not looking for just heads when we flip a coin. We want to have specific distributions of outcomes within a set number of events. So not all events are going to be, not all of the 7 events are going to be albinism versus normal. We have much more complexity in the requirements. And so we're going to use a more coherent way to bring them together into a definite probability. And so this is kind of a strange equation that has exclamation points in it. But it's actually not that impenetrable. So n, we know n from statistics as like population size. In this case, it is population or sample size. And in this case, it is the number of events that we're concerned with. So number of events. And an event basically in probability is, you know, a specific occurrence, like a thing that we're looking at, such as the birth of a child with a specific skin trait. So n is the number of events. And when we are looking at these events, we're largely looking at a dichotomy. Like we're looking at one of 2 things happening. And we talked about it a little bit earlier, but in probability, we express like antithetical outcomes to a specific event as P and Q. For example, when we flip a coin, it'll either land on heads or tails. And because those are our only 2 options, it is going to happen 100% of the time. The probability of one event happening plus the probability of its opposite happening equals 1. It will happen all of the time when you add them together. So if P is the probability of a specific event happening and Q is the probability of its opposite happening, we can look at that within the question. What are the 2 like, possibilities that could come from this event?
But was that a question?
Yes.
repeat it? Sorry, there's something.
So looking at this event, what are the two possibilities that could result in this event?
Either the child. Are you talking with albinism or with the tails?
With this specific example.
Okay. Either the child has like albinism or doesn't.
Exactly. The child is either albinistic or not. Normal, it's called. Nothing wrong with being albino. And whether or not you assign albinism to P or Q is arbitrary. Like albinism could be Q just as long as you keep the following straight. So we have these two things that can happen, and there's a specific probability that they will happen. And so the way that we integrate these probabilities into an equation that is all about like specific numbers of times that these individual events are happening is by using the variables S and T. So S is the number of times that the event associated with P happens, and T is the number of times associated with the event that Q happens. I didn't word that properly, but whatever. And it can be hard to keep these straight, but the way that I do it is that I know that Q is always associated with T just within this equation here, because you don't want to accidentally put S with Q. That would just destroy your math. I remember that Q is with T because QT is like you're saying the word cutie. Very fun. So T is associated with Q, S is associated with P. So if P is the probability of a child being albino, and S is the number of times we're looking for that the event associated with P happens, the number of children that are albino, what would be S?
S would be like how many children have albino.
Yeah, and how many would that be?
We 3.
Yeah, so that's 3, and it was also articulated that 4 are normal. And so now that we have, oh, actually we didn't articulate that. So we have S and T, but the one important thing that we haven't mentioned so far is what is P and Q? We know like what events are associated with P and Q. P is associated with an albinistic child and Q with a non-albinistic child. But what we need is to find the actual value of P, like what the probability actually is. And that must be in the previous question. Hopefully, maybe, please. Humans' albinism is a recessive trait, so we can assume, let me find a color that I haven't used. We can assume that if albinism is recessive,
Because like I know in class she just used like a heterozygous cross for like both parents.
Oh yeah, the reason that she did that is found here in the question. Can you identify what about this question indicates that they're both heterozygous?
It says that the parents are carriers.
Bingo. Parents are both carriers, and I did a very poor job of explaining that earlier, but it's pretty intuitive. They're carriers because they carry that recessive allele, but they have a dominant allele that keeps it from being expressed in their phenotype. And so we know that's the cross of the parents. The parents are both heterozygous, so we're going to do a nice little monohybrid cross. That makes sense, right?
I have a question.
Yes, what's up?
This is probably very stupid, But the word carrier, does the word carrier mean heterozygous, or does it just mean that you have the allele?
Heterozygous. Well, actually, there isn't really a difference. Someone who is a carrier is heterozygous, and we know that because hetero means different, zygous means, or it's referring to the alleles. So they have two different alleles. This is a single organism, and that single organism has two different alleles, one dominant and one recessive. And being a carrier is like an extension of that. Like they
Oh,
carry one kind of allele.
oh, like what about like if it was like, what happens if they like are like homozygous recessive? They still carry the
Yes, you are definitely right. But in that case, like when we think of carriers, well, when I think of carriers, I usually think of like the carrier of an infectious disease, like they're asymptomatic, but they still carry it within them. You wouldn't really describe someone who's openly coughing and hacking up spit and stuff as being a carrier. Like they're obviously sick, but someone who isn't showing the symptoms of that disease, but is still infected with it, that's someone we'd call a carrier.
Oh, okay. Sorry.
Yeah, so it's kind of arbitrary, but um, actually, no, I think you make a good point. A homozygous recessive person definitely does carry um, two recessive alleles, but um, yeah, we reserve the carrier phrase for someone who's specifically heterozygous because it's not evident in their phenotype. But it can still obviously contribute strongly to the genotype of their offspring if they happen to pass on the
Yeah,
recessive allele versus the dominant.
I got it. Thank you.
You are welcome. And so when we do this cross, we know that familiar 1:2 to 1 genotypic ratio that results from the cross. And that is such that 1 is homozygous dominant, 2 are heterozygous, also carriers, and 1 is homozygous recessive. So going back, the probability of being albino. So in this case, because albinism is recessive, only this genotype would create an albino phenotype. And because there are 1, 2, 3, 4 possible offspring that results from this cross, and each one is just as likely to occur as the other because it's random whether or not the parent will give this one versus this one, because they're all just as likely as the other, and there are 4 possible offspring, then it is 1 out of 4 that the offspring of this cross will be albino. And so P, the probability associated with the event of albinism, is 1 out of 4, 0.25. And by extension, because we have these two genotypes that both translate into a normal, in quotes, phenotype, that is 3 out of 4, or 0.75. And so now we have all of our numbers that we need. We have the values of P and Q, the values of S and T. We know N, because N is, does it make sense where N comes from?
Yeah.
Yeah, N is like the population or the total like sample of events that we're going to be doing in the first place. So we have all the numbers, and now we can just very nice and cleanly plug it in to our equation here. And so when we do that, we get that N is 7. We know that S is 3, and we can just plug it in really fast. Let me do that. So 7 factorial divided by S is 3 factorial. and then we multiply it by p, which is 0.25 to the fifth power, or no, that's not a 5, that's an s, to the third power, sorry. And we then multiply it by q, which is 0.75 to the fourth power. Oh, dang it, I messed that up. So we need to, sorry, one second. That's a lot better. Do this times it. And for that, I got the probability equaling 0.173. Does that look familiar?
I tried to put it into my calculator, but I got like a different number.
What'd you end up with?
I got like 6.22.
Like a whole number?
Yeah.
It could be because when I tried it the first time, I got 99.66, and that's just because I made a error in my calculator. So it could be that.
Cause like that was like happening to me a lot during class.
Mhm,
So
yeah,
like I asked the guy next to me how he put it in, and then he did it first time, and I got the answer.
yeah. I also have a problem like using a calculator at all. So that could actually be a problem on the exam. So we can definitely talk more about that. I don't think I can like broadcast my calculator to Zoom right now, but in class we can talk about that because that is probably going to be important. Whether like how to find the factorial button and using parentheses and stuff. Because yeah, like we know what we're doing, and it would suck to like get the question wrong just because of a calculator error. So as long as that's the only problem, I think we're good. Everything else is sensible, right? Where we got the numbers, what they all mean.
Yeah. So like, would you say for to get the probability, we always have to like actually do the crosses out by hand?
Oh, definitely. Unless it's explicitly given to you, then yeah, unless the probabilities are given, because you know P and Q are probably the most important part of the equation. Uh, if you're told about two people mating and producing offspring, you're going to have to do the cross because that is what informs, you know, the P and Q, the probabilities that make up the most important part of the equation.
All right. And like, I'd also have like a question like, how are we set up a dihybrid cross?
Perfect, no problem. I'll just use this as a whiteboard. So dihybrid, we talked about it a little bit earlier. And it means a very specific thing. It refers to a very specific thing. So di means two, hybrid is like two different things coming together. It's an amalgamation. So a dihybrid refers to, you know, hybrid, two different things like a heterozygote. It's a hybrid between a dominant allele and a recessive allele. And we have two of them. We have this hybrid and this hybrid. So we have our di, two, and hybrid, different. Oh my God. And so that's what we're looking at. We're looking at two different heterozygous conditions. And this A is at one specific locus. Locus 1, and this B is at another locus, locus 2, And what do we find at different loci on a chromosome?
Like different traits.
Yeah, what codes for these traits that we have?
Alleles.
Yes, exactly. And what do alleles represent?
Like dominant and recessive genes.
Genes, perfect. Ah, that was perfect. Thank you. I couldn't have said it better myself. We, at different loci, find alleles that represent the genes that inform our phenotypes. And so when we have a dihybrid cross, so this dihybrid, these two hybrids, because we're looking at a hybrid at two different loci, this is the genotype of one organism. Obviously, it's like larger than that. We have so many genes. So when we're looking at ourselves and we're talking about cystic fibrosis, like our entire genotype isn't made up of like this genotype, but that's just what we're looking at because we're only concerned with the genes related to cystic fibrosis. And in a dihybrid cross, similarly, we are, in the case of the dragons we looked at earlier, only looking at like scale color and egg light properties. So if this is one organism that we're seeing, looking at two of the genes in their genome, a dihybrid cross, therefore, is a crossing between two dihybrids, AABB, crossed with another organism. So this is one organism, and here is another. And that's how we set up our cross.
What is like they be put in like grids?
Oh, that is what we are trying not to do because it's just so much. We don't really want to invest all the time in the world because, preferably we're going to be doing this on an exam and we don't really want to have to write all of this business out. So what we can do instead of doing the actual Punnett squaring is to remember these nice little ratios, 9 to 3 to 3 to 1, where from the, sorry, let me get that back. Never mind. From the result of this cross, because the parents, the individuals in these cross can say give this gamete and this gamete, creating an AA BB, or just by chance, they could give this gamete and this gamete, lowercase a, uppercase b. Because of that, they can create different distributions, different amounts of offspring that display certain phenotypes. And that's what we're really looking at here in this Punnett square, is that if you were to do this 16 times, if you were to make this cross happen 16 times, we see that a certain phenotype arises on 9 of those times. And it's because of these crosses, the parents happen to have given a capital R, capital Y versus a capital R, capital Y. Or they could have given a lowercase r, lowercase y with a capital R, capital Y. And so just based on this happenstance, we come across a different distribution of genotypes that translate to a well-defined ratio of phenotypes. And that is the nice and pretty 9 to 3 to 3 to 1. Let me bring that back. Oops, I messed it up. Anyway, yeah, 9 to 3 to 3 to 1. And that is where 9 of the individuals, let's say if we're looking at this roundness versus color locus, 9 of them are going to have sorry, 9 of them are going to have the at least one dominant allele at the first locus and at least one dominant allele at the second locus, just based on the chance of whether or not the individual gave a capital R with a capital Y, a lowercase r with a capital Y, 9 of the 16 individuals that result from this cross will have this genotype with at least one dominant R and at least one, that shouldn't be a D, dominant Y. And then 3 of the individuals will have at least one dominant allele at the first locus, but 0 dominant alleles at the second locus. Another 3 will have 0 dominant alleles at the first locus, at least 1 at the second locus. And then 1 of the 16 individuals, based on probability, will have no dominant alleles at the first locus and no dominant alleles at the second locus. And these all correspond to specific phenotypes, which will be like given to you in the problem that you're asked.
All right, thanks.
You're welcome. Did that answer your question?
Yeah.
Awesome. Oops, that's not it. Yeah, I think I talked about everything that I wanted to. And we went through a little bit of extra stuff. I don't have anything else. And if you guys don't have any other questions, then I think we're good for this week. And obviously you're welcome to email us or ask us questions in the Discord or talk to us in or after class. We're learning assistants. We're here to assist in your learning. We don't have any other purpose. So yeah, ask us your questions. We're here to help. But for now, I think we can be done.
Thank you.
You are so welcome. Thank you guys for sticking around. And for those of you who watched the recording, thank you. And I will see you in class on Tuesday. Bye, guys.